Yes.
Perfect. Sounds good. So yeah, thank you so much, Eugenio, and all the organizers really for giving me this opportunity to present this work in such a cool venue. I am very excited to be here. So as Eugenio was saying, I'm a scientist at Regeneron in New York area. And for today's talk, I would like to present some of the work that I did as a postdoc also at Regeneron. And the project, this study was recently published in Nature Communication at the end of last year. And so the title of this presentation of this work is IL-33 expands plasma cells, disrupts germinal centers, and increases autoantibody production. Okay, so basically, as a brief introduction, I just wanted to point out that IL-33 is a cytokine that is released upon tissue damage. So it can be released by structural cells. And IL-33, after it's being released, it can activate different immune cells that express the IL-33 receptor, or also known as the ST2 receptor. And this receptor is expressed in different cells as it's here shown in this cartoon. But interestingly, B2 cells, or more commonly known as classical B cells, don't express this ST2 receptor. And in addition to that, there has been several studies that show that IL-33 and also the soluble ST2 receptor can both be increased in the serum of patients that have different autoimmune conditions, including rheumatoid arthritis, lupus, or myasthenia gravis. So we thought that these findings could suggest a potential role for IL-33 in inducing autoimmunity and or inflammatory damage. But this project really started with this experiment, so with this observation. So what we did here is that we systemically deliver different cytokines using a technique called the HDD, or hydrodynamic DNA delivery, that allows us sustained and high systemic levels of the cytokine that we wanna study. So we did that at day 1. At day 7, we harvest the mice, and here what we did is we looked into the spleen of these mice. And here I'm showing you the total plasmablast and plasma cells in the spleen of the mice. And as you can appreciate, when mice receive HDDL33, we saw a significant increase of plasma cells in the spleen of those mice as compared to the empty vector, which is the negative control, but also as compared to other cytokines that we studied. We looked at the isotype of those plasma cells, and we saw that that all isotypes were significantly increased, and that also correlated with increase of circulating antibodies in those mice. So after seeing this observation, we thought we questioned ourselves, how are these plasma cells being generated when IL-33 is overexpressed? And this is of particular interest because, as I was mentioning in the introduction, B cells and plasma cells don't express the ST2 receptor. And so we wanted to confirm that also ourselves, and we did that looking at ST2 positive cells in the B cell compartment, and we see that at baseline, or with the empty vector as a negative control, we don't see ST2 on the surface of B cells, but also after the induction of IL-33, there's no increase of this receptor. So really confirming that this receptor is not expressed on B cells, as compared, for example, of T cells, where we do see an induction of this receptor on T cells. So this has to be mediated through an indirect effect. And so in order to understand a little bit more about the mechanism by which this overexpression of IL-33 induces this plasma cell expansion, we repeated our experiment, but before the HDDL33, what we did is that we prophylactically treated the mice either with an isotype control as a control, and or with anti-CD4 or CD8 depleting antibodies, or anti-IL-5 antibodies. So here I'm going to start showing you the data with the anti-CD4 and CD8 depleting antibodies. The first thing that we saw is that there was no impact of the in mice treated with the anti-CD8. So CD8 doesn't seem to be particularly important in this mechanism. However, in mice that were prophylactically treated with an anti-CD4, and then we did the HDD-IL-33, we saw that there was so we see an increase, for example, of these IgG1 plasma cells after the HDD-IL-33. And interestingly, when mice were treated with the anti-CD4, we see a decrease of IgG1 plasma cells. And we saw the same for IgE plasma cells. However, when we did the same analysis, but looking here at the IgM plasma cells, and it's the same for also IgG2A plasma cells, we don't see any impact of the anti-CD4 treatment. So that suggests that the expansion of IgE and IgG1 plasma cells is actually CD4 T cells are contributing to this expansion, but T cells cannot contribute to the expansion of all plasma cells when IL-33 is overexpressed. So we did the same exercise, but looking at what happens in mice that were previously treated, so prophylactically treated with the anti-IL-5. And we saw that in this case, IgG1 plasma cells and the same for IgE plasma cells were not impacted by the anti-IL-5 treatment. And in this case, interestingly, IgM plasma cells were impacted by the anti-IL-5 treatment, and it was the same for IgG2A plasma cells. So it seems that, so as a conclusion, we realized that there was probably different pathways that were contributing to this increase of this expansion of plasma cells, and depending of the isotype, CD4 T cells or IL-5 were contributing, or at least partially contributing to this, expansion of plasma cells. And to confirm the implication of the CD4 T cells and the anti-IL-5, we repeated the same experiment, but here we added an additional group where mice were treated with anti-IL-5 plus the anti-CD4. And so here we looked again at total plasma cells in the spleen. And what we saw is that while the anti-IL-5 and the anti-CD4 only partially reduce the plasma cells after the HDD-IL-33, probably because they don't impact all the isotype, it was in mice that were treated with the anti-IL-5 and anti-CD4 where we saw that the phenotype was more or less restored compared to the empty vector, and suggesting that that potentially both pathways are sufficient to induce this plasma cell expansion. And so I have here a cartoon that kind of summarizes a little bit what we are thinking about the mechanism by which the overexpression of IL-33 systemically leads to this expansion of plasma cells. So we think that there's at least two mechanisms that are pathways that are participating in this expansion. On one side, we have T cells that express these ST2 receptors that contribute to the expansion of IgG1 and IgE plasma cells. And on the other side, we think that there's other immune cells that express the ST2 receptor that are capable of releasing IL-5 when they are activating, that are probably contributing to the expansion of IgM and IgG2A plasma cells. So after this, the second question that we had was, okay, so we see an expansion of plasma cell, but what happens at the level of the B cells? And so one of the experiments that we did in order to answer this question was to directly look in the spleen of these mice by immunofluorescence. And so we are staining B cells with the anti-CD19 in yellow. We also have a marker of IgD in red, so cells that are CD19 and IgD positive will be naive B cells. And then we have an anti-CD3 marker in purple that would show the T cells. And then finally in white, we have BCL6 staining that would indicate germinal center cells. And so I hope that you can appreciate that compared to the empty vector control, when mice receive IL-33 HDD, we observe a disorganization of the splenic architecture. And we try to quantify that by looking at the percentage of the area of the follicles. And we saw that in mice receiving IL-33 HDD, we observe a decrease in the percentage of the area of these follicles. showing that there is an impact of this HDDL33 directly in these germinal center structures. And then we zoom in directly in one follicle. And as you can here see and appreciate in the control, we are able to see the T cells, we have able to see the naive B cells, and we are also able to see the germinal center in white. And we can clearly see that with IL-33 HDD, this is the structure of this germinal center is disrupted. And we actually saw, interestingly, that there was a decrease of the BCL6 positive staining, as you can see here compared to here, but also we in the quantification. So this is quantifying the number of BCL6 positive cells in comparison with the total of splenic cells. We saw a significant reduction of these BCL6 positive cells with the IL-33 treatment, confirming the loss of the germinal centers. So knowing that with IHDDL33 we are losing this germinal center structures, we wanted to understand what could be the impact of that. And as a reminder, and since we are today in a germinal center session, so I just want to remind you that germinal center structures are very important structures for the adaptive immune system. They are responsible for different things. First, they are important to increase the affinities of the antibodies that are produced after an immunization, and that's through somatic hypermutation that happens in the dark zone of the germinal centers. But also those structures are important to enforce the self-tolerance by avoiding the emergence of autoreactive B cells, and that happens in the light zone of the germinal centers. So we wanted to understand, okay, we are disrupting the germinal center, so what happens? What happens then? So I'm going to show you first experiments looking at the affinity of antibodies after an immunization in the context of overexpression of IL-33. So in order to do that, what we did, quite simple experiment, we immunized the mice with MPKLH with alum. So this is a well-known T-dependent, germinal center-dependent immunization that is used in mouse models. And 7 days later after the immunization, we gave to mice either HDDL33 or the empty vector as a control. And then 7 days later, we harvest this mice. So the first thing is that we looked at the B cell population in the inguinal lymph nodes, so the draining lymph nodes, and we confirmed that in mice receiving IL-33 compared to mice receiving only the empty vector, there was a reduction of the germinal center B cells. However, there was no impact of other subsets of B cells, including NP positive, so the antigen positive memory B cells, or also no impact on naive B cells in the inguinal lymph nodes. And then we were interested in understanding what and how is this antibody response in those mice receiving the IL-33. So we were interested in the quantity of the antigen positive antibodies, but also in the quality of these antibodies. And so in order to do that, we can do in the lab two ELISAs in parallel, where we use where we coat our plates either with MP2 BSA, so where we are going to be able to capture only high affinity antibodies, or we coat our plates with MP27, which basically is going to detect all antibodies, so the high affinities and the low affinities. And finally, we can do a ratio of NP2 to MP27, and an increase in this ratio indicates an increase of the affinity of the antibodies. So what we saw first was we look at the antigen, so NP positive IgM, and we saw, and we were not surprised to see that, an increase of this IgM, so NP positive IgM titers, probably because we are seeing this expansion of plasma cells and no difference in the ratio. But interestingly, when we look at the IgG1 response, what we saw is that the ratio of MP2 and to MP27 IgG1 was decreased, suggesting a decrease in the affinity of the IgG1 antibodies produced follow the immunization. So that suggests that with the overexpression of IL-33, we do see that there is an increase of antibodies, so there's more quantity, but the quality of these antibodies is probably is probably reduced with in mice having high levels of IL-33. So that was for the first part, where we confirm in a mouse model after an immunization that the affinity was disrupted in mice receiving high levels of IL-33. And then on the second bucket, we also wanted to understand what happens in terms of tolerance in those mice, since we know that those germinal center structures are important to enforce this peripheral tolerance. And so in order to answer this second point, what we did is we went back to our wild-type mice. So those are wild-type mice, naive mice, they have not been immunized. And we gave HDD-IL-33 or empty vector as a control, and we looked in the serum of those mice 7 days later. We looked for anti-double-stranded DNA antibodies. So those are autoantibodies that are detected in patients with autoimmune diseases, such as in lupus. And what we saw is that in mice receiving IL-33 HDD, there was an important and significant increase of anti-double-stranded DNA titers. The majority of them were IgM, as you can see here. And there was a trend, although not significant, of increase of IgG1. So we also looked for other autoantibodies, and you would find it also in the paper. But what we saw is that in these mice receiving HDD-IL-33, we were seeing an increase of autoantibodies in circulation, suggesting that that high levels of IL-33 can disrupt this peripheral tolerance, and we were able to find more autoantibodies in circulation. So the next step was to try to understand what could be the consequences of this increase of autoantibodies. And we wanted to do it in the context of a mouse model of autoimmunity. So these NZBWF1 mice, they are genetically modified, and they develop spontaneous autoimmunity that with features that resemble human SLE. So in those mice, they will spontaneously develop autoantibodies. So they will already have anti-double-stranded DNAs. They will have immune complex deposition, proteinuria, kidney damage, and those mice also show a premature death. So those mice are already predisposed, and they are already they will already develop a pathology. But we wanted to understand if in those mice we do see an impact of high levels of IL-33. So what we did in those mice is that we gave either the empty vector as a control or IL-33, and then we follow the mice for survival. And what we saw very interestingly is that mice receiving, so NZBW mice receiving IL-33, we saw there was a premature death compared to mice that receive only the empty vector. So there was an acceleration of the pathology. And we were interested in understanding why those mice were dying faster. And what we did is we collected the urine of those mice, and we look at the ACR ratio. So this is the albumin to creatine ratio, and basically indicates kidney pathology. This is looking at 4 weeks after the HDD, and we saw that in mice receiving IL-33, there was an increase of the ACR ratio. So that shows and should suggest that those mice were having more kidney pathology, and that was probably the reason why those mice were dying faster. And finally, the last piece of data that I wanted to show you is the role of IL-33 in lung. So all the data that I have shown you so far is data in the context of systemic delivery of IL-33, so with very high levels of IL-33. It was an interesting model for us to understand mechanism, to understand how this IL-33 can potentially trigger more pathology in mouse that are already predisposed to autoimmunity. But IL-33 is an important cytokine in airway diseases. We know that in patients with asthma or COPD, the levels of IL-33 in the lung, and not in circulation, but in the lung, they are very high. And so we wanted to try to model that in mice to try to understand what is the impact of IL-33 in the B cells in the lung, to try to understand this concept in a more physiological relevant context. So in order to do that, we decided to locally expose the mice with IL-33 directly in the lung. So here we are not working anymore with the HDD system. It's just we intranasally deliver either saline as a negative control or recombinant IL-33 in the mice for 4 weeks. And then we harvest the mice at day 28. We harvest the lung and we looked again at the plasma cells and plasmablasts in those mice. And what we saw, it was very interesting to see that we were able to observe similar things that what we were seeing when we systemically deliver IL-33. So we saw an increase of plasma cells in the lung tissue. And again, also an increase of plasmablasts. So plasmablasts and plasma cells were increased. We saw that there was an increase of antibodies in the lung. And when we specifically looked at autoantibodies to see if we were also able to observe this break intolerance in the tissue, we also observed that in mice receiving recombinant IL-33, we observe an increase of anti-double-stranded antibodies, confirming the relevance of these findings in the lung. And the last experiment that I would like to show you for today is an experiment that we did using our Hassel's mite mouse model. So this model helps us to recapitulate a long chronic inflammation model. In this model, so mice are exposed with house dust mite for 3 times per week for 15 weeks. So it's a quite challenging model where you can see chronic inflammation, where you see so long eosinophils, neutrophils, so like a mixed inflammation. We do see a lot of cytokines also that are increasing the lung, so mixed inflammation. We observe persistent and severe airway remodeling. So it's really a model where we can really see a chronic inflammation, and the mice are quite severe and sick by the end of the experiment at week 15. So we have, so if you are interested more in this model, we have also other publications describing a little bit more this model. But in this case, what we wanted to understand it was what could be the role of IL-33 on the B cell side in this model. And so what we did is that starting at week 4, so therapeutically, and at week 4, those mice are already quite sick, we started the treatment with either an isotype control or with an anti-IL-33 antibody, and those mice were treated twice per week, and we harvest the mice at 15 weeks. And so what we did is we looked for autoantibodies in the lung again. So here's looking at anti-nuclear antibodies in the lung tissue. And what you can see is that in mice receiving house dust mite, a little bit after 4 weeks, but really at 15 weeks, we observed that these autoantibodies are increased in the lung of mice receiving house dust mite. And very interestingly, we observed that mice treated with the anti-IL-33 treatment were showing a decrease of these anti-nuclear antibodies, suggesting that in this model, the increase of autoantibodies is at least partially dependent on IL-33, and like that confirming the role of IL-33 in this break of tolerance and in these autoantibody productions in the models in this model. And with that, I would like to conclude. So I have shown you today using different ways of delivering IL-33, so either systemically or locally, we observe an increase of plasma cells. In the spleen, we observe an increase of all isotypes. In the lung, I didn't have time to show you, but we saw an increase of IgG1, IgM, and IgA in the lung. And when we look more at the mechanism of and also the consequences of these high levels of IL-33 systemically, we observe this loss of the germinal center, and then triggering a low affinity antibody production in the context of immunization, but also importantly, increasing autoantibodies and breaking the peripheral tolerance and this can also lead to an increased pathology. And with that, I would like to thank you for your attention. Just a reminder that this study was published quite recently in Nature Communication. And finally, also, I want to acknowledge